Welcome to the NIHR Dementia Researcher Midday Lecture webinar. I'm Adam Smith from the Program Director at the Office of the National Director for Dementia Research at UCL, and I'm delighted to welcome Dr. Sam Moxon. Hi, Sam.
Hi, thank you very much for inviting me on. It's good to be here.
I've followed Sam on Twitter for quite some time, and I was delighted when he agreed to join me on the Chatathon event that I organised last month. For those that haven't already watched that, you can go to chatathon.uk and find lots of content from over the 12 hours that I talked to 56 different researchers over that time. After the Chatathon, I have to say, when it came to Sam's discussion, I was left wanting more. So I was delighted when he agreed to join us today to record a webinar for us, which gives him a lot more time to go into the details of his research and his work at Manchester. So, as I said, Sam is a research associate at the University of Manchester, and today he's going to talk to us about his research on working in bioprinting and in a webinar entitled Building Brains for Medical Research. The talk will be around 20 minutes, and then we've allowed about 10 minutes for questions at the end. Anytime during the talk, you can use the Q&A button at the bottom of your screen to post your questions. And then when we get to the end, I will put those to Sam on your behalf. We're also recording today's lecture, so if you have to drop out for whatever reason, don't worry too much. This will be available sometime, probably tomorrow, on our YouTube channel. So thank you very much for joining us again. And Sam, over to you if you'd like to perhaps share your screen now.
Obviously, when I spoke to you, Adam, before we agreed to do this talk, I talked a lot about bioprinting. But what I wanted to sort of outline today is the fact that you cannot just go straight into bioprinting, and actually it's quite a long process. So I thought I'd take you through some of the steps we've taken in moving in towards these really interesting models of the brain for studying things like dementia. And specifically, I come from a background of a bit of cell biology. So I did a degree in genetics. But then since PhD, moved into a field called Biomaterials Science. And that essentially means engineering materials to interact with biological systems. So it's about taking a material that is able to interact with biological substrates, cells, tissues, and integrating the two for a medical application. So for example, a good example of a biomaterial is a dental composite, which a dentist would put into your tooth to aid healing. But it's not just about using biomaterials to regenerate tissues. We can also use them to make models of tissues and study things like diseases and drug screening. And it's particularly useful in something like neurodegeneration research. A lot of the experiments that you tend to see with cells around diseases like Alzheimer's involve growing them on plastic dishes. And really, that's not representative of what actually goes on in the brain. So in the brain, we've got this three-dimensional network of neural cells all interacting with each other and all surrounded by an extracellular matrix. And that's really important to their function. It's as important as the cells that surround them. The materials they interact with have a big impact on how they behave. But instead, we're creating these two-dimensional flat cultures on plastic. And it's not ideal for studying something like Alzheimer's disease. Because if you think about the pathologies of Alzheimer's disease, you tend to have two main hallmarks. You have misfolding of tau causing tangles and discrete deposits of amyloid plaques. And it's thought that the plaques precede the tau tangles, and it's all linked in this feedback loop. So then a patient will start to have buildup of amyloid plaques, which should then lead to inflammation and tau phosphorylation and tangles. And you get this slow degeneration of the brain over decades. And then you start to see the onset of symptoms like short-term memory loss, long-term memory loss, confusion, hallucinations. It just gets worse and worse as time progresses. And the structure of the brain is integral to this process. So what I'm trying to get across is if you grow cells on a two-dimensional plastic substrate, and let's say you're looking at amyloid aggregation in Alzheimer's, and you're trying to model that for the purpose of trying to then screen potential therapies that can prevent amyloid beta aggregation and plaque formation. If your cells are producing a lot of amyloid, say for example, cells from an Alzheimer's disease patient that you know has a mutation that causes them to produce excess amyloid, Any amyloid that's produced in a two-dimensional culture is just going to simply dissipate up into the media. So there's no time or opportunity for it to aggregate into plaques. However, what we know and what we've shown is that if you encase your cells in a three-dimensional biomaterial, specifically one that's designed to replicate aspects of brain tissue, you can actually then start to promote amyloid beta aggregation and potentially start to see the formation of discrete plaques. And then that gives you the opportunity to look at the process that underlies plaque formation, look at the potential to treat it with certain drugs, but also look at how that can have a knock-on effect on other cells as well. So by putting our cells moving away from 2D to 3D culture, we can start to build something that looks more like the disease that we're trying to study. So what we tried to do in our lab is combine two pieces of technology. The first is induced pluripotent stem cells. So essentially you can take somatic cells from a skin biopsy from a patient with Alzheimer's disease, and through a stepwise process of introducing different pluripotency factors, you can then revert those back to a state of pluripotency, i.e. you've got a pluripotent stem cell that can become any cell type. If you then differentiate that into a neural subtype, say a neuron or an astrocyte, what's interesting is the mutations that are present in the neurons in the brain of that patient are also present in the cells you've got growing in the lab. So you can almost get a cross-section of what is going on in that patient's brain. So we take our induced pluripotent stem cells and combine them with a class of materials called hydrogels. And these are, as the name suggests, gels that are comprised mostly of water. In the case of what we do, we're talking 99.5% water, but they can still form these self-supporting structures. And what I'll touch on later is we can also print these into really precise structures. But before you do that, you need to know a little bit about the material itself. And it's important because the content of your hydrogel, both from a chemical and physical point of view, so what it's made up of and how stiff it is, has a big impact on how the cell behaves. And actually we know that if we take a population of stem cells, we can have as much control over what cell type they differentiate into simply by changing material stiffness as we can by giving them specific chemical cues. So with a hydrogel, we can essentially put a cell into an in vivo-like environment in vitro. So in the lab, when our cells go into a hydrogel, if it's designed appropriately, it can mimic aspects of the tissue that the cells are supposed to be residing with in the body. So what we really do is to design a suitable hydrogel is go back and look at the tissue itself and look at the matrix that surrounds the cells within that tissue. So what we did in our lab is take a look at the primary components of brain extracellular matrix. And one thing you see in different regions of the brain is a polysaccharide called hyaluronan, which is linear, it's carboxylated, and it interacts with other proteins inside the matrix, and it sort of holds the matrix together. So what we wanted to do was create something that was similar to that, a linear carboxylated polysaccharide interacting with proteins. Now, hyaluronan itself is extremely expensive. It's hard to get hold of large amounts and very difficult to work with. So we basically went back and looked at materials that are structurally similar, because in an application like this, you don't necessarily have to make an exact brain replica. You just need to make something that gives the cells the impression that they are sat in an appropriate matrix and will promote them to carry out the normal functions. So we chose a polysaccharide called alginate, which you can get from seaweed, but structurally it's very similar to hyaluronic acid. And basically interacted that with the easiest protein to get hold of, which is type 1 collagen, and created this hybrid material matrix where we've got collagen fibers interacting with a polysaccharide mesh. And stiffness-wise, it's very similar to brain tissue. So what's interesting is if we embed our iPSC-derived neurons, so we take our induced pluripotent stem cells and take them through a differentiation process to make cortical neurons, so neurons from the cortex. If we put them within this gel, we see this formation of 3D neural networks. And it's all to do with how the matrix is designed. So because we have the collagen in the matrix, the cells can interact with the collagen via integrin binding sequences and form this three-dimensional complex network. And not only form it, but also mature it as well. So it's one thing to have a neuron inside a gel that looks like a neuron. It also has to behave like a neuron. So we measured a couple of different things. One of the things we saw that was interesting is presynaptic physical proteins starting to traffic down the neurites towards the synapse, the longer we kept the cells in the gel. So they're starting to prime themselves for electrical activity. And I'll show something a little bit later that demonstrates what this activity looks like. And they also start to become more branched as well. So we start to get this really busy three-dimensional neural network, and we're starting to grow these sort of mini brains. And I won't talk too much more about this study. This is published now, and the reference can be sent out if anybody's interested. But this represents the first layer of complexity that we can build. But in actuality, the brain isn't just one cell type in one matrix. And actually, the brain comprises of a couple of different components. And there's a wider structure that we call the Neurovascular Unit. Because the brain is so active, it requires a lot of oxygen to keep it fueled, and therefore has this vast blood supply that penetrates through the brain. And it forms this neurovascular unit where we've got interactions between cells of the brain and cells of the blood vessels that spread through the brain. And what's interesting is in diseases like Alzheimer's and other dementias, it's not just the brain cells that are affected. We can see a negative impact on the blood vessels as well. And then that can create a much more aggressive feedback loop where degeneration of the brain leads to degeneration of blood vessels, and things that shouldn't be able to get into the brain can leak into the brain, cause further damage. And it's just this really big feedback loop. So what we want to do is build on from just looking at a single cell type and start to look at that structure as a wider composition. And in particular, we want to take our three-dimensional principles and move it out towards something like the blood-brain barrier, where in a normal case, the blood-brain barrier specifically controls what goes in and out of your brain to stop anything toxic getting in there. But in diseases like Alzheimer's and vascular dementia, we see that break down and things start to lead in. And again, we come across the same problems when looking at blood-brain barrier studies. Again, we're looking at studies where cells are grown on plastic with single cell types in isolation, and it doesn't reflect the fact that either directly or indirectly, all these cells are communicating with each other and in very specific matrices. So what we thought would build on from our simple gel of our neurons and start by just trying to coat the surface of our gel with a layer of microvascular endothelial cells. So these are the cells that line those blood capillaries and vessels and control what goes in and out of the brain. And so can we add another cell type that then gives us a more complex model. So similar to before, we've got our hydrogel, our brain component, which contains within it our stem cell derived neurons that form these beautiful 3D networks and express all the right markers. And as I alluded to earlier, form these active networks where they are signaling to each other. So each flash you see here is a change in calcium potential, which suggests that they're actually able to communicate with each other and form this electrically active network. So they're actually actively communicating with each other spontaneously, as you would expect to see inside the brain. But not only do we have our neural component, we now start to have our vascular component. So on top of that same gel, we have our brain microvascular endothelial cells forming this barrier that is selectively permeable. So we are starting to get this blood vessel-like quality to the top of our co-culture. And what's interesting as well is we can tweak the conditions to get the best possible function. So we can optimize it to get the most functional barrier to the point where we can see these are tear readings which tell you the resistance across the barrier. We can get values that match that reported in vivo. So we're starting to get something that's like the resistance of a human blood vessel. And what's interesting is if we look at a cross section of this, the neurons that we place within our co-culture form these beautiful networks and spread out through the entire 3 dimensions of the culture. This is a sort of a side-on view from a confocal microscope. We can see that our neurons, which are highlighted in red, are spread throughout the entire gel. So they're not just sticking to one location, they're actively spreading out and forming this network. But also our endothelial cells stay on the surface, form this barrier, and don't start to migrate down into the gel. And that's important because you don't want that. You want to have that barrier forming over the top to be maintained, because the second they start to migrate down into the gel, that barrier can become leaky and you've not got a good representation of what a human blood vessel would normally be like. But most importantly, it's okay to make something like this, but you then need to try and find an application. So one of the things we're looking at the moment is modeling disease mechanisms. So for example, in diseases like Alzheimer's and vascular dementia, you get a lot of damage from reactive oxygen species. So we're looking at whether or not we can induce damage to this, and then looking at how that can potentially be recovered. It gives you the opportunity to look at a disease mechanism and then try and solve that disease mechanism. And that's a good way to screen for potential drugs. But another thing I touched on earlier is the difficulty in getting things like amyloid beta plaques, which we know are so critical to disease progression. One thing I'm not going to touch on too much because it doesn't relate to a great deal to the bioprinting aspect I'm going to talk on next, is the fact that we're now starting to get evidence that these green sort of blobs here are labeled with a amyloid beta antibody. We see that if we pick the right matrix and grow our cells under the right disease conditions, so for example, if they've got a mutation that pushes amyloid beta synthesis, or if they've got an enzyme that is supposed to degrade amyloid beta inhibited, we can start to see these plaques forming. And so what another thing we're quite interested in is if we have our neurons within our co-culture starting to form these plaques, does then that have a negative impact on the endothelial cells? But on top of that as well, we're not just looking at neurons and BMEX, we're also looking at getting other cell types in there as well. Astrocytes, pericytes, all very important. And this is where I think the stuff that excites me most comes in. And that's the 3D bioprinting side of things. So a 3D bioprinter is what it sounds like. It's a 3D printer that prints biological substances. So the one on the left is actually one that we have in Manchester. It's made by a company called Regen Hue. As you can see, it's quite large, but it gives you the opportunity to do lots of interesting things. So these images on the right are just a couple of representations of what's been 3D bioprinted. I should state that's not a functional ear and that's not a functional heart. This is just showing the kind of architectures that can be made. But specifically, why I think bioprinting is important for something like the brain is because the brain itself, as I mentioned earlier, is complex. It's different matrix compositions, different cell types, and they're not all just bundled in together. They're placed in very discrete locations and layers. So all the gels I showed you before is essentially mixing cells with a gel, leaving it to set and not controlling where any of those cells are located. But in the brain, we have these very discrete layers with different cell types and different materials sometimes. And it's very difficult to recreate that just by making a gel by hand. But what's interesting, and something that we developed when I was doing my PhD, is if you take a bioprinter and print your materials when they're in a liquid state, so before they form their solid gel, and print them into another matrix that supports them and behaves a little bit like amniotic fluid does in a developing fetus, you can actually layer up these materials when they're still in liquid phase. So you're basically laying up liquid on liquid on liquid, and then solidify it and create these very precise layered structures. And it gives us the potential to start to create this layered structure that you see in the brain. So this is what the process looks like. We've got our amniotic fluid-like support bath, which is this sort of cloudy stuff. And the blue is our bioink. This is actually the matrix I talked about earlier, which is the mix of alginate and collagen. And the first video, we're printing something that's similar to a blood vessel. And the second one is a simple lattice. And the reason we print a lattice is because it gets us to a point where we can see how high resolution we can make our filaments, so how thin we can get them, how far we can space them apart. And that gives us an idea of how thin we can make each layer. And we think we're at the point where we can make our layers between 150 and 250 micrometers thin. So we can make very precise structures. The purpose of the blood vessel relates more to the stuff I talked about with the blood-brain barrier. And we want to work towards having a structure where we can have our blood vessel-like gel that contains our neural cells within it. But then on the inner lumen of that, we will have our vascular type cells. And therefore, we've got something that looks like a cross-section of a blood-brain barrier. And what's promising is if we put our neurons into these 3D printed materials, they still survive and start to form processes. It's very difficult to image these, so it's not the best quality image, but we are starting to see neurite formation, and we're working on a better way to try and image these at the moment. But ultimately, we want to work towards a position where we can integrate multiple different cell types to build cell crosstalk across the brain in both a healthy and disease setting. And work towards investigating relationships between neural and vascular dysfunction in Alzheimer's disease. So the stage we're at is we've developed our bioinks. So these are the things that you put the cells in to print. That took a couple of years to get something that worked well for all our cell types. We can also make these 4 different cell types from iPSCs. We can make our endothelial cells, the pericytes, astrocytes, neurons, and make them quite reliably. And we can embed them all within our bioinks and see a positive response, i.e. they don't lose their function when they go into the gels. So the next stage is to start to work towards building up these layered depositions of different cell types. And by doing that, you can look at specific configurations. So for example, if you wanted to look at the interactions between pericytes and astrocytes, you could put them in layers side by side. And it gives you that opportunity to really customize in a way that you can't do when you look at, say, an in vivo model where you've got the whole organism and you can't pick apart those different cell interactions. So that's where we're heading. We are opening a brand new facility at the University of Manchester later this year called the Royce Institute. That's going to have a dedicated 3D bioprinting lab, which I am hoping to be a part of, because it would be really interesting to see if we can really take this to that next level and build these things that actually look more and more like mini brains. But that's where we're at the moment. I'd just like to finish by acknowledging the funders and everyone who's helped me along the way, particularly going outside of the university, Professor Alan Smith and Professor Liam Grover, who helped me design this bioprinting process in the first place, and everyone at Manchester who's got me to where I am. Hopefully, in another few years, we can show you some even more interesting data.
Fantastic. Thank you, Sam. That's fascinating. And I'm just going to remind our audience now, if you'd like to ask questions, if you're watching on YouTube, you can post that in the comments box below the video you've just watched. And if you're in Zoom, you can use the Q&A buttons at the bottom of the page. And I will put those questions to Sam now. That's fantastic, Sam. So the new printer that you're getting your hands on, you said that's next year.
Well, hopefully later this year. We've got all the equipment. It's just waiting for the building to be finished. I mean, it looks finished, but it's just getting all the lab space prepared. We've actually got the printer up and running. It's just in a different facility for now, and it will move across to the Royce Institute when that opens. And it's built inside a Class 2 cell culture hood as well, so everything is completely sterile. It removes the chance of infection, so you can actually make cellular structures instead of just making, like that ear I showed you earlier was just plastic. We can actually make proper structures now. We
have got a question come in. Danielle Jones asked, in future, how do you believe this approach may be implemented to study neurodegeneration? I can see the challenge there, because is this a case of, if are you going to need to have 3D bioprinting facilities in every lab in the country that wants to use this mechanism, or is this something that you can produce and then keep alive long enough to pass on for the study of those to happen elsewhere?
Yeah, so that's a good question. In terms of transporting a bioprinted structure, I've actually done that in my PhD with one for osteoarthritis. So we made an interface between bone and cartilage, and that was transported for about, I'd say, only about 30 minutes across from the building in the University of Birmingham over to the hospital. So transporting long term might be difficult. The good thing is, the printer I showed is very expensive and very high tech, but you can actually get printers for much lower cost and are much more easy to integrate into a lab setting. You don't necessarily need to be an expert. But the good thing about, and this is just going to sound like an advert for the Royce Institute, the good thing about this is the whole point of that is to have it as a collaboratory hub. So if you are interested in a 3D printed model, instead of going into that alone and having to start from scratch, you can consult with people at the Royce Institute, tell them what it is you would like to do, and get expert advice and expert input on how we could do that. And the other thing is, you don't necessarily have to bioprint. The model we showed you earlier, where it's just 2 cell types, is not bioprinted, and it's much simpler to work with. It just depends on what questions that you want to answer. And if it's quite a complex question, sometimes a simpler model is actually better to use, because if the biology is complex enough, you don't want a really complex biomaterials model on top of that to further complicate it. It just gives you the ability to do very customized things should you wish to.
Yeah, that makes sense. And if this can be kind of scaled down, and if you prove the concept in the way that you are doing, you can see how then that could be transferred. Thank you for the question, Daniela. Next, we've got a question from Phoebe, who asks, what has been the contribution of iPSC to research into amyloid-related tau mutations that was not already known from murine post-mortem models?
Yeah,
Is that your area?
Not specifically, but it's a good question, and it always comes down to the same thing with iPSCs. The difference between looking at iPSCs and post-mortem is massive, because obviously post-mortem you're looking at end of life, you're looking at basically severe Alzheimer's disease, whereas when you make iPSCs and make neurons, what you make is actually very fetal. And so even just getting a mature tau sort of proteome, I suppose would be the right word, basically getting the right subforms of tau to be produced to correlate to mature neurons in iPSCs is very difficult. And actually, Selena Ray's group in UCL showed that it can take years to get that to happen. But it then gets back to the question of, is that necessarily what you want to look at? We know that with Alzheimer's disease, by the time someone gets diagnosed, it's already too late. And there are a lot of mechanisms that precede those symptomatic changes. And so if we're trying to find some of those really early stages, are we better off looking at something that is perhaps more, not fetal, but somewhere between the two? And so iPSCs could potentially contribute to that. But I guess it's not trying to say that one way is better than the other. It's a case of using everything we've got, and iPSCs are just one part of that. So a question that you might find iPSCs useful for, you won't find perhaps in vivo models for, and vice versa. It's all about, I think, using as much data as we can get from as many techniques as possible. But in terms of tau, it's still quite difficult in iPSCs to get a mature tau phenotype.
Yeah, actually, that kind of takes us on nicely to some of the questions that are coming up. Next question is from Brian, who asks, if you print the different cell types in different layers, will they be able to move to different layers? That might eventually result in basically the same mixing of cell types together and seed them in the hydrogel. You can see that. Do they move between? Yeah.
Well, it depends on how you design your material. If you want them to move, we can make them move. If you don't want them to move, we can keep them where they are. Porosity is a big thing. That dictates how much they will move. Stiffness also. What we see is over a period of 60 days, they don't tend to move too much. It all depends on the material. With the osteochondral one, which is it's a good one to mention, because we actually made an implant that we stuck into a piece of human tissue, ex vivo, I should say, for ethics reasons. We weren't actually trying it on patients, but we cultured this two-layered structure with two cell types in each layer for about a month, and each cell type stayed in the region that we put it in. It all depends on how you design the material. And so if you don't want them to move, you can design the material so that they can't move. But if you would like a little bit of migration, it is also possible to stimulate that as well. It's all about the material.
I suppose the key here is getting the message out to the different labs that are working on, cells in dishes in this way, that this opportunity is there to look in this and see if it'll enhance the research that they're already doing.
Yeah, and it can sometimes seem a bit inaccessible, because if you try to do this from scratch and look at the materials that a company will sell you, they're going to charge you hundreds and hundreds of pounds for something that isn't necessarily very good. So just finding someone in your Department of Pharmacy or somewhere like that, someone who knows a bit about biopolymers is much more useful, I think, than going to a company, and you can get something that's a bit more bespoke to your needs as well than spending hundreds of pounds on something that doesn't necessarily do what you want it to do.
I'll take this opportunity to mention that Sam is on Twitter if anybody wants to catch up with Sam on there, and I know he tweets regularly about his work and highlights various papers and things. He's at Dr Sam Moxon. So do give him a follow on Twitter. We've got a couple more questions. Julianne Clement asks, did you see mature plaques in your model strained for thioflavin or similar things? And did you look at the effects on tau, phosphorylation and aggregation?
We're actually doing that right now. We're currently looking at tau phosphorylation. I believe that we're actually running blots this week. I say this because I don't actually run the blots. In terms of the mature plaques, we're also looking at that. We've stained with, the stain I showed you earlier was 4G8, which is just an amyloid antibody. We've stained with BTA1, which is a thioflavin derivative that has a much higher affinity for amyloid beta, and potentially seen some evidence, but we're still not 100% on that. So we're also looking at a couple of other different antibodies as well to try and get a better profile of what it is that we're seeing in those aggregates. And in terms of promoting that, we've looked at lots of different things. One is things like the APP Swedish mutation, whether that produces more amyloid, and also inhibiting certain enzymes. I'm not sure how much I can say on that at the moment, but inhibiting certain enzymes and seeing if that produces more plaques and that kind of thing. So yeah, that's all stuff that we're working on. But that doesn't necessarily require bioprinting, so I've not touched on that too much today.
Fantastic, thank you, good question. Next question is from Johnny, who asks, is amyloid generated in these 3D models if the iPSCs are taken from someone with Alzheimer's? If so, how long does it normally take to see a beta accumulation, and does it need any kind of trigger to accelerate the process?
Yeah, so again, that's a tricky one to answer it, but we do see amyloid aggregation, not in the material I showed earlier, in a different material, and I think it takes about 3 or 4 weeks. It depends on the cell density that you put into there. Sometimes we've seen it with no trigger, which is strange. We're still trying to work that out, but more often than not, an Alzheimer's related mutation definitely helps. You have to pick your mutation quite wisely, because you want something that's going to produce more total amyloid, as well as perhaps changing the ratio of 40 to 42. But also, like I said, the work we're doing on inhibiting a certain enzyme to see if that also produces more amyloid. So yeah, I think some of the studies where they have 2 or 3 mutations are a little bit overkill and not necessarily clinically relevant, because you won't find a patient with more than 1 mutation. But we're still trying to get a good spectrum of actually what kind of mutations you need to start to see these aggregates and when. All we know is we do see some, and we're still working out exactly what's going on there.
Makes sense. Thanks, Johnny, for the question. And looks like this is our last question. Last chance. Anybody, if you have a question, please do post it now. Julienne again asks, if the cells don't move much, could you possibly use bioprinting to print different layers of a cortex? Yeah,
something I'm very interested in. Obviously, I think you're probably talking very high resolution by that point, and so it's getting it to a much even smaller scale than we're on now. But yeah, this is something that we are interested in, and something that I'm talking to another researcher about as well. Again, I'm not sure how much I can say, because it's not work that I'm taking the lead on. I'm just assisting. But yeah, it is something that we're very interested in trying to do, because if you can recreate the cortex, it gives you the opportunity to do some really interesting stuff.
Fantastic. Thank you, everybody, for those questions. Oh, last one. Sneaked in there. Well done, Phoebe. Could you use the technology to model the combinatorial effect of SAD risk genes, ApoE, TREM2, etc.?
Yeah. Again, you've got the freedom to plug in whatever cell type you want. We've actually got an ApoE mutant iPSC line. We've not looked at too much yet. But yeah, you can. The good thing is, as long as you've got a robust system, you can plug in different cell types and look at contributions. One thing I will say is these things take a lot longer than you would expect, because particularly things like if you're relying on imaging and you've got these 3D structures, you often need to do a lot of optimization before you can start to get results. And things like getting protein out of these structures to do things like Western blots took a little bit of time to get that right. But yeah, you can plug in different cell types. I'm not going to make huge leaps and say what that will show you, but it's definitely something that's worth trying.
Fantastic. Thank you, everybody, for those questions. I have one last one, Sam, is so one of the things that our office has been looking at is encouraging people potentially from other professions and other areas to consider bringing their skills to dementia. Is there some other technology, some other sector, some other engineering sector out there that if they were to bring their skills to this, it would accelerate this path of research, do you think?
Specifically with the bioprinting, or just in neurodegeneration?
Yeah, probably, I guess with the bioprinting.
Yeah, I mean, process engineering, a lot of process engineers, someone we work with on this is a guy called Marco Domingos, who's an engineer. Well, I hasten to say he's an engineer, because he now knows a lot about biology and about all this kind of stuff. But yeah, someone who knows a lot about these processes and how to optimize them and how to make the best out of, someone who can understand what is an appropriate processing strategy for the material you're interested in. I think it's important to get engineers and biologists and clinicians all talking together, because but the biology alone is very interesting. But if you want to do something complex, you're always going to need an engineering aspect of it. Even if it's things like a microfluidic chip, you're going to need an engineer to produce that for you.
Yeah.
So somebody who understands from the other perspective, materials and processing is always useful, particularly if you want to build something very bespoke.
Absolutely, you can see that bringing different professions in to kind of tackle this challenge. I think bringing some minds together would be fantastically helpful. Thank you very much, Sam. We're very grateful for you agreeing to share with us today. As I mentioned before, you can find Sam on Twitter. His details are at Dr. Sam Moxon, so please do go look him up. You can also see the chat we had previously on the chatathon.uk website, where I talked to Sam there with Dr. Louise Serple, Professor Louise Serple. We don't have a date finalized yet. However, our next webinar will come in November with Dr. Daniel Erskine from Newcastle University discussing his research on Lewy body dementia neuropathology. So keep an eye out on our webinars page to find out when that's going to be. The recording from today, as well as all future webinars, can be found on our website, which is dementiaresearcher.nihr.ac.uk/webinars, and also on our YouTube channel. It's youtube.com/dementia researcher. While I've got your attention, I also want to remind you that the Race Against Dementia Fellowship Call is now open at the moment and accepting applications through to the 20th of January. You can find out more about that in our latest podcast, where you can listen to that wherever you get your podcasts, and they'll also be holding a webinar to take questions and offer any advice on the 16th of November. Details can be found in the calendar on our website. I think anybody who's listening to this and working in your field, this is exactly the kind of fellowship call that would be right for you. I'm also delighted to let you know that myself and my collaborators recently successfully applied to start a new Alzheimer's Association iStart professional interest area called the PIA to Elevate Early Career Researchers, called PIAs for short. I'm very excited to be the first chair of this PIA. So if you're an iStart member, please do go log into your iStart account and you can tick the box to become a member of that group. We've got a lot of exciting things that we're hoping to work on over the next year. Last of all, if you'd like to join us and present your own webinar, please do let us know. You can find us on Twitter at dem_researcher or drop us a line using the contact us page on our website. Well, that's a lot of plugs at the end. Thank you very much again, Sam, for joining. Thank you.
Yeah, thanks again.
All our audience for taking time out of your day to watch this webinar and for all the questions you posted. Thanks very much.
Thank you.